Assalamu alaikum and welcome to our R Genomics tutorial. I am Dr. Hamad Ismail. Today we will talk about the data containers and annotation hub in Bioconductor. This is the third lecture of our Bioconductor series. In the first lecture we talked about the installation, and in second lecture we talked about BioString's annotation hub and some other related parameters. Bioconductor is a powerful R-based tool which can be used for the sequence data analysis like RNA sequencing, chip sequencing, or next generation sequencing. The best thing about Bioconductor is it's a free open source software. It means you can use it for your data analysis and you can publish it free of cost. As our today's tutorial mainly focus on the data containers and annotation hub. As we know that all the biological data are basically in the experimental data form, which contain the raw measurement, just as gene expression value. The second most important thing is the metadata. Metadata means sample information like age, sex, treatment, IDs, etc. And the third level is annotation. Annotation is external contents which refer to the pathways, gene IDs, and conserved regions, and so on. To process all such data, we need to process the raw data. The raw data first goes to the pre-processing. Pre-processing means filtering, removing the ambiguous values and sorting out the main values. And then we place them in the data container. Then from the data container we go to analysis, and from the analysis we go to the results. The key concept behind this tutorial is that the data container ensure consistent operation across the dataset, which means all the pre-processing are placed in a particular data containers, and these data containers are constant and all the pre-processing undergo to the same data containers and the analysis become homogeneous. So let's start in the R. First of all, you need to install the R. This is the very first command. We check that either your R is properly installed or not. It command says if required. If and required mean if BioManager is required, install it quietly. It quietly means it will not pop up any message, it will install, and then installation package will start. If it is already installed, it will not initiate any installation. Let's say I need to run this command. Simply select the whole command and press enter. It will evaluate, and let's say it's saying that Bioconductor version 3.2 is already installed. Next, the second thing we need to run this database is we need to install three packages. Which number one is BioBase, number two is ALL, and number three is HGU95 database. Simply select this command and press the run button. It will start installing these packages. These three packages, BioBase is basically the expression database, ALL is acute leukemia example dataset, and HUG95 is microarray based dataset. These are three example datasets on which we are going to work. After the installation of these packages, we need to load these packages. For this we need to load via the library. Select this command collectively or one by one, just press enter, and all these packages will be loaded. Once we have loaded the library, first of all we are going to see the example data like this command data, and under the data function we have used the ALL. When we run this command, it will load all the data. Let's say you can see this is over here. These are all the data loaded here. When you print this all, it will show you all the information that what kind of data set is present and what information is present in it. As I told you earlier, this is acute leukemia example dataset. It contains 128 samples. These are the 1 to 6 to 5 features and 128 samples. and it contains approximately 128 sample names, and data set seen 21 time total, like code diagnosis and any other thing. And this is annotation about HUU91. So this is the information we get from this all the data set. Once we have loaded all the data, after that we can use different function. For example, we can use the function of express. Express is basically represent the experimental measurements, and then we can use pdata. Pdata is basically the sample information. Feature names represent the gene information, and sample name present the sample IDs. Let's say I am using the express all, and in the large parentheses we used 1:4 comma 1:4. 1:4, it means the first position and the fourth position. This is the first position and the fourth position. It means it will represent first 4 gene and first 4 example. Let's run it. When we run it, you can see the result over here, and it will show you the first 4 sample. Express and run. Now it is showing you the first 4 sample, the number 1 sample, number 2 sample, number 3 and number 4. Similarly, if we run this command, it is showing you the feature names, all the features name which are present in the sample 1 until 127. You can find out the features you are interested. Then we can see the pdata. Pdata present the sample information. Run this command and it will give you the information about all the samples which are present. You can see a big file over here, which means number 1 is code, then the diagnosis type, the date when the diagnosis was made, the sex, male or female, age, then BT level, permission type, CR level, data creation, and so on. These are all the information present over here. The next thing is, if I from the acute leukemia cell I put a $sign. $sign means you are going to fix something from the complete data set. In this case, as I know my parameters, my column names are, if you see in this data, the column names are diagnosis, code, sex, age. If you want, you can fix something. Like here I fixed the sex. You can also fix the age, you can also fix the CR, you can also fix the remission. Let's say I fix the sex and press run. It will give you the value of each column and each value, that either they are male or female. They have printed only the sex column. Same way you can let's say we type it here, and instead of sex we make age, and then press run. Now it is going to print the age. Similarly, you can print any other parameter you like. In the same way, like we did for the sex, we can also sort the data in the form of pdata. Like we use the same pdata all command. This is the same pdata all command, but we are going to fix it for the sex. As we put the $sign, it is going to fix for the sex. Run it and it will give you the information only about the sex column in all the pdata. So in this way you can access the data inside the gene expression set, and you can look for the specific expression you are interested in. If you are interested in some more specific expression, these are some examples, like in ALL. Here I want to explain one more thing, that let's see about this one. This is the data. Here I have placed 1:10 comma space 1:5. 1:10, these first parameter represent the row and this represent the column. If you run this, it will give you the results. about the parameters. About it's saying it's 5 total values out of 21. It say these are the first 10 rows from the data and first 5 rows from the column. If we only run this one, we have added nothing in the row column. It will take only 5 values from the column rows. Select this and press the run. It is selecting the 5 and it is only selecting. If you run this way, it will collect the first 10 rows of each data set and it will give you the result. It say these are the 28 values. When you run this one, it say these are 25 values. Or simply you can create another vector like for the row I have placed nothing, and for column I have created another vector. The C symbol is represent the column values, and I said 3, 2, 1. It will choose the values in the same order which I have placed, and then click run. It say these are the values. It has given us the 3 values which I have placed it, 3, 2, 1. So you can do lot more in the topic, in the idea. Like you want to do some sequence analysis, you can find out a specific age, you can find out a specific data set, and so on. Our next section is consist of annotation using HUG9, which is a microarray data. Here we are going to make another variable and we are going to use the features names, and we are going to place the data set acute leukemia data set, and we are interested in first 5 values, which means we are taking the first 5 names from the acute leukemia data set, and we have created a variable with the name IDs. Just run it. You can see the IDs name are over here. Now we are making as list. As list is basically the R built-in function, which is a base function its core purpose is to construct the data in the form of R-friendly database. So we are getting the data from an online database and we are making it user-friendly for the R. And here we are interested in HUG90 for increased, and here this is our name, and then these are the IDs. These IDs are the same vector we have placed in it. Simply run this command and you will get the result. You can say it has taken the first 5 values and placed in the R friendly format. This is what we were going to do. This is what we want to do, so that we can process and read it clearly in the R friendly format. Now let's move toward the next step. Now we take another example for RNA dataset for some rise experiment. Let's suppose we want another package with name Airways. Airways is another package for RNA database. Simply install this package, run this package and start. It will install the package. If it is only installed, it will pop up with a message. If not installed, it will install. After installation, load the package Airways using the library function, and then load the library for genomic ranges. Run it. Then we want to see that what kind of data is present in the Airways. Simply use the data in bracket, use Airways and run the command. After running the command, simply you can see Airway data in here. Or you want to see in console, you can simply run select the Airway and run the command. It will show you all the data of the Airway. You can see this is the class range summarize experiment with dimension 8 into 63677, and these are the counts, and these are the gene IDs and sample information and many more things. So you can use this Airway package to analyze different kind of data sets. Now if we want to do a specific information about the Airways like column data, simply run this command and you can see how many columns are present and what information is present in these columns. It will give you the results. Similarly, the column names, if you want to see the column name, run this command. These are the names of the columns used in this data set. Similarly, assay names, what assay was performed, these are the assay, like counts, this was the assay which was performed. Then we are interested in the counts, okay? Like in database of airway, what count stands for. Run this command and you will see the result. Here these are the results. You can see this is a big data about the counts. These are basically column names, these are the row names, and these are basically their counts. You can see that these are different kind of data you can see. Row range, this is another function, and you are interested in airways and you are interested in row number 1, just simply select this command and run it. You will see that what kind of data, like in rows, these are the X values and these are the negative strands and column IDs and exon names, and the ranges are present. These are basically different kind of exon. Approximately 17 ranges are present with 2 metadata. Metadata is basically the information other than the main key information. If you are interested to see the start of airway, like airway is another database and you want to see that what is the start of airway or very first entry, like first 10 entries, what they say, simply select this command and press the run button. So it will give you the first 10 entries of data, and these are basically the base pair code. This is, it says that its length is 1, and these are the sequence, like it is starting from this, and it is ending from this. These are the ranges, I ranges of these airways data set. Next, if you want a more detailed analysis, you can create I ranges and G ranges as we discussed in the previous section. In this case, I am going to create a variable or vector with the name gr, and I am going to create a G range with the sequence name is 1, and ranges I ranges start from 1 and end at 10 to the power 7. And just create this variable, run. It has now created the variable gr formal class range, and these are the data from the gr, or simply subset by overlaps. You want to create a subset and find out the overlaps in this airway, then you can use the function of subset by overlaps. This is the main function. In this main function, we are using the package of airway, comma, and the vector or variable we are using to use it the gr, which we have just generated. Now run this command and you will see the result in the console. It says it range randomized experiment, dimension are 329 or 8, metadata is 1, assay is counts basically, and row names are ENGS, this and this, and row names are 10, and these are basically the sample information. So you can find out different overlaps between the airway package and the gr. Gr is our vector. Using these commands, you can find out or inspect or visualize the large data set in a very quick and simple ways. The next part of this tutorial is how you can access the public data. For accessing the public data, we can use the function of geoQuery, which is getGeo and getGeo supplementary files, or we can also retrieve the annotation programmatically using the Biomart. These are the packages which we can analyze. These packages and these functions help us to retrieve the data which is present on NCBI or assemble or some other base, and they help us to retrieve and download on your local directory easily. The main idea is between that we choose the series of GSC, then we make the sample at GSM level, and we perform at specific platform GPL. They will find out the use the GSE and make the samples, and then from that platform we can analyze that data. So let's have a look on this. So first of all, we need to install the package GeoQuery. Geo is Gene Expression Omnibus. So simply first of all, we need to install this package using the Bioconductor manager. Once it is installed, we can use to use their function. There are two basic functions. One function is get geo and then get geo supply file. First of all, our package has been installed, and then we will load the library of GeoQuery, and then we can access the data using get geo. Get geo download the process data, while the get geo subfiles access the raw data. So this is the basic difference. It is this command will get the process data, and this will download the raw data. So first of all, we run this command, and it will download the data. To access this data, you need to have an active internet connection. It will download the data directly from the NCBI website and will give you the information. Let's see, this is the data. This is GE11675 series metric, and it contains the 6 samples, and these are the information about this data. This is PubMed ID, and this is the annotation hub, which is GLP 8300. Now the second information, now we will run this command, and it will upload all the data which is required. It has now downloaded the data in our folder. It is saying that that data has been downloaded, the raw file has been downloaded in this folder. Desktop, lecture number 3, and GES and raw. Let me show you the data. This is the lecture number 3, and you can see it has downloaded all the raw data in this format. You can process or use this data in the next steps. If there is something happens and you are this link GeoGet data is not working, you can simply go to the download link. Let's suppose this is the data which I have accessed from the NCBI website, and you can download this data and place it in your folder. After placing it in your folder like this one, I have already placed this in my folder. This is the data I have placed. Then placing in this folder, you can use the directory to get wet. Either you are in the right folder or not, it will give you information that yes, you are in the right folder, and then you can access this data using this command. So this is an alternate way to access your data if this above command is not working. But most of the case, if you have a stable internet connection and if the NCBI server is not down, you will get your results. If you simply run this command now, GST, and it has accessed the data, you can see that the data is present over here, or you now simply can print the gset run. It will give you all the information. So in this way you can also get the raw data information over here. Other way to access the public database is Biomart Basics. When you install the Bioconductor manager, it automatically installs the Biomart packages. If not, you can install the Biomart package using this command. In this command, you can simply copy this Biomart and place in this folder. The structure is already prepared and pasted. Now, when you will run this command, it will install the package. Once the package is installed, you can load the library of Biomart. The Biomart library has been loaded. Next, list marks. We can see that what kind of list are available in our mart. This is the basic function of biomart package. Run this command. It will give you the list of all the packages, like it says ensemble genes are available, mouse strains are available, ensemble variation available, and ensemble regulations are available. Now, next step, what I want to do, I want to say that I create a new variable with the name mart. In the mart, I say use mart. Use mart is the function of biomart. In this biomart, I have accessed the ensemble genes. So I put the ensemble in the inverted commas and bracket closed. When I run this command, select this command and run it. The mart has been generated over in these variables. This is the mart package and it contains all the information. Next thing is that another function, next thing is which is list dataset. It's saying that list all the datasets which are present in the biomart. In this dataset, I am interested in the variable mart which I have just created. Now, select this data, list mart over here and press done. It will give you the list of all the packages available over here. You can see these are all marked lists present in your genome database. You can select any parameter from here for analysis, for cutting of DNA sequence, for finding the overlaps or reducing it, finding the genome ranges, or any other function. These are all public available datasets. And the next step, I am interested in the mart, like use the dataset of Homo sapien gene and in ensemble. So I use another function. I created another vector ensemble and I give it the direction that use the dataset. Dataset is also function of the biomarker, and I said in it use the dataset from the Homo sapiens. Homo sapien ensemble gene. Homo sapiens, bracket closed, inverted commas closed, then comma, and in the mart. When you place the mart in it, it will produce the function about the mart. So after completing the command, run the command, it will come up with the dataset. I just left this command over here, comma, mart, and bracket closed. So ensemble is ready. You can now simply print the ensemble and see the result. Object of the class, it is the object of class and it contains biomarker. In the ensemble, I am interested in two things. First thing is the function which is list attributes. I am interested in the attributes of the ensemble. And the second thing is the list filters. The list attributes give you the functions like what homologous pairs. Oh, sorry, I need to run it again. These are basically information which are features and names and IDs are present which are present in ensemble gene ID. And the second thing is list filter. If I run this command, it will give you the information of some specific filters which can be performed like Panther ID, PFAM IDs, residue ID, print IDs, ProSite pattern IDs, and so on. There are a lot of data available. You can see when you see the data in the public repository like Ensemble, like NCBI, there are a lot of information is present. But we are interested in some specific information like superfamily, tiger form, or something else. You can type the information in the list and can find the gene annotation in that particular sequence. The last part of this tutorial contains some information about S3 and S4. Now what is S3 and S4? Both are the let's say programming algorithm in the R package. S3 is the let's say it is simple but powerful. It has flexible and interactive section. It uses some generic function. Mainly its functionality depends on the argument. For example, it can use the argument like plot and methods. In other ways, S4 methods are more formal with definition of the classes. Like Bioconductor packages are mostly based on the S4 package, and they have some validation of type and naming convention. Like in my descriptor, use the genome description. Like here it's the plot and only the plot. Like here we are specific in genome description. Both methods are used in R, but in the Bioconductor packages mostly the S4 functions are used. Why we use S4 in our Bioconductor package? Because S3 packages are very bad at validating the type and naming convention. They do not do that. Their inheritance work but depend upon the input. We need to provide the input. These problems are not present in S4. However, these are some complex structure, but things are clear as we are going to deal with the complex DNA sequences, RNA sequences, or protein sequences. So we need to be very specific to differentiate between the different sequences. So the best method used in R for Bioconductor is S4 method. So let's check either our Bioconductor is using this S4 method or not. It is very simple to check either our functions are S4 based or not. As we know from the start of this tutorial, we were using the database of acute leukemia model. So we want to check the class of this model. Simply select this function and press run. It's a expression ATR package and it is bio-based package. Now the next function is if this method is S4. Is for that the command is S4. Type this command and type the name of the model or biobase. Run it and it's come as true. If it come as true, it means that definitely it is S4 based. If it comes as false, then it means it is not based on S4 model. Now if we want further differentiation, like say here it said it is expression set. Now we want to see the get class of the expression set. Simply use the function of get class and type expression set. Copy this command and press run. Yes, it extends with that it is directly and version biobase by class is set distance by 2. You can see here these are basically class expression package biobase and these are slot. Yes, its expression is different level. Then we can see the method as data frames. You can show the method, simply run and it will show you the method. What method is used to create the database, They use array, bitmap, data frames, genome range, IPOS ranges, relays, list. These all methods were used to create this kind of packages and data set. And you can also use the get method to see either data frame or data set has been used or not. Simply run it and you will get the answer. Yes, it is showing you the data frame of the matrix, that how they created the data frames in the data matrix, and these are the condition. If condition has been applied, else condition has been applied, row names, row column, and all the structure has been displayed to you. And at the end, either we can validate the object or not. For this purpose, we use the function of valid object. For the valid object, we use the function or use the database ALL acute leukemia cell. We can run this command and yes, they say yes, we can validate the object. And these all commands are basically the validation of that data set, and it can be only applied to S4 model. If some packages are not S4, if they are S3 packages, then we cannot validate these commands and these results will not be shown in that particular database. So it confirms that all of our data is S4 data. So in summary, in today's lecture we have talked about expression set, which are basically the classic containers, and then we talk about summarized experiment, which contain the modern container along with the genomic ranges, and then we talk about the annotation and how we can access the public data which are essential for the analyzing the genomic sequence, and then we talk about the S3 and S4 objective system, and we talked about how we can validate that either this object is S3 or S4. This is all about today. If you have any question, please ask in the comment section. If you liked our video, please like and subscribe. Thank you so much for watching. Allah Hafiz.